أهلا وسهلا فيكم، أهلا ومرحبا جميعا فردا. المناعة في زمن الكورونا. معانا ضيفة رائعة اليوم، معانا الدكتورة جوان بينوك من جامعة مانشستر. أنا منتظر رفيقتي في هذا الحوار، الدكتورة طرفة التركي. دكتورة طرفة، المايك مفتوح لك الآن، تسمعيني دكتورة؟
السلام عليكم.
وعليكم السلام ورحمة الله.
I'm so happy that you are with us today.
Thank you very much for the invite.
دكتورة طرفة، تعطينا بريف عنك لو سمحتي بالعربي.
أنا دكتورة طرفة التركي، أستاذ مساعد في الإيميونولوجي في قسم العلوم الطبية التطبيقية، و I'm a PhD graduate of University of Manchester, and I'm so happy today that we're going to talk with Joe about vaccines and tests in COVID-19, because Joe is a passionate immunologist and an extraordinary scientist, I think. What do you think?
Yeah, I agree. 100%.
Thank you.
Joe, how are you?
I'm good, thank you.
I'm guessing you understood every word I said right now.
Most of it.
Most of it. Well, 11 years of knowing me and knowing most of your Middle Eastern students, I'm sure you have. So where in the UK are you now?
At the moment, I'm in mid Wales with my family, enjoying lockdown and some unseasonably warm weather in the UK.
So where, for the people who haven't been to Wales, tell us maybe one or two, let's say three, kind of trivial pieces of information about Wales.
Well, in the context of coronavirus, it's important to know how many people we've got in Wales. So Manchester's a central point for many people who are listening, I think. Greater Manchester has about 3 million people as a population, and Wales has 3 million people as well, but is considerably larger. We also have a very nice set of mountain ranges that's grouped together as Snowdonia National Park, but most of them are not really at the global scale of mountains. The highest one is about 3,000 feet, but it's very beautiful, a very long coastline and some very friendly natives as well.
I've lived in Aberystwyth for years, as far as you remember. How far is Aberystwyth from where you are?
About an hour's drive from where I am. So I'm south of Harlech, which is a really famous castle in Wales. I'm right at the bottom southwest tip of Snowdonia, near the coast.
Wow. Well, it's beautiful. I've been there. I don't miss a chance to come and visit Wales anytime I'm in the UK. All right, so we've done the pleasantries. And the mic is yours.
Okay, well, welcome everybody. Mushraf asked me if I would give an overview of the current understanding of tests and vaccination strategies for coronavirus or COVID-19 or SARS-CoV-2, however you would like to describe it. So I'm just going to share my screen and give you a few slides of the overview of the virus and the pandemic as it stands at the moment, and then give you a few introductory slides on tests and vaccinations, where we're up to at the moment. And when I say we, I mean the royal we. Introduce myself more accurately now. So I'm just going to share my screen here. So here we go, hopefully.
Yes.
Right, whoops. Oh, that's not right. Hang on. 1 second. One second. I thought I'd set this up correctly, sorry. 1 second, here we go. Right, so this is me. I'm Joe, or Dr. Pennock, from the Lydia Becker Institute of Immunology and Inflammation at the University of Manchester. And just to give you a bit of context, this is Manchester City Centre. This was our highest building, is no longer, and this is where we are based and where Tafa and Mushraf would be very familiar with in the Faculty of Biology, Medicine and Health. What is most important in the context of this talk really is that we're right next to one of the biggest hospital complexes, teaching hospital complexes in Europe, which is just down here, just next to the buildings where we do a lot of biological research. So the Lydia Becker Institute, one of the primary themes that we have is barrier immunology. And as you know, coronavirus is, and so we have researchers within the Lydia Becker who are already looking at the immune responses, particularly the blood immune responses to coronavirus. And we can talk about that more at the end of the talk. So what do we already know about coronavirus or COVID-19? As of last 2 million cases to date. These are confirmed cases with via PCR tests. We have nearly 145,000 deaths so far, and 57,000 patients are still in a serious or critical condition, and this number is likely to rise. So this graph here, which is taken from the ECDC, so the European CDC, gives you an indication of the number of cases globally in graphical format. So you can see in Europe here, we have the majority of cases, and I'll show you a map in a minute. This is closely followed by Asia and then America. So even though America daily has the highest number of new cases, actually cumulatively, it has fewer cases than Europe at the moment. What else do we know about this coronavirus? It's the 7th known human infective coronavirus. It falls in the genus of a beta coronavirus and is in the same genus as SARS-CoV and MERS-CoV, which I'm sure you've heard of both of these and have caused pandemics in their own right. And this is why a lot of the discussion around the immunology to coronavirus really centers on what we already know about vaccinations and immunology in the context of these other two types of coronaviruses. The genus name is SARS-CoV, so I'm going to use these two things interchangeably here, so COVID-19 and SARS-CoV. But importantly, there are no licensed vaccines yet for human infective coronavirus, and this includes SARS-CoV and MERS-CoV. And there's an awful lot of debate about why this is, and we could talk about this in a political context at the end of the talk. But just for now, there are no known vaccines. What we do know about it, though, is that it's through contact and through droplets from coughing and sneezing. And what I think is really interesting, so this is a slide I've used for many years in my immunology lectures when I teach the clinical immunology students, the master's students at the University of Manchester, is that when you look at the migration patterns globally, so this is from the UN Migration Agency, and really depicts airplane patterns. And you can see there are hotspots here in China, Europe, and America. And this is the main, this is by people rather than a number of plane journeys. And you can see the three main business centers, really, I suppose, of the world. And then when you compare this to the hotspots of coronavirus, it very much maps onto this migration pattern. So this is really how the virus has been able to spread so rapidly. And as I showed you on the previous graph, a hotspot of infection now is in Europe, where we have many hotspots of more than 100,000 cases, particularly in the UK, France, Spain as well, and Italy, of course. So when we think about this as an infection, and we compare it to other pandemics that have been known and recorded, why do we consider that we're in the middle of a public health emergency? Now, for obvious reasons, there are many deaths in a very short space of time. It has spread incredibly quickly in a matter of weeks, really, across the world. But when we compare it to other pandemics, we can make some really interesting comparisons. So you'll have already heard of the influenza pandemics of 1918 and 1957, and then in more recent memory, in 2009, the H1N1 virus. And when we look at the fatality rate for influenza, it's usually less than 3% and more often less than 1%. But the influenza season is long, which means that these deaths occur over a long period of time. Before, there was public health measures in place, testing, serology, and then more recently, these lockdown initiatives that have been happening across the world. The number of deaths were incredibly high. So, for instance, influenza in 1918 led to 20 to 100 million deaths. And the reason that range is so broad is because the actual number is not known. Interestingly as well, the average age of fatalities in 2019 was 28 years old. So it really hit between 20 and 40 years in that case. In 1957, I couldn't find out the average age for the fatalities, but you can see that far fewer people died from this infection. That doesn't mean that fewer people were infected, and we'll talk about that a bit more in a minute. And then the first coronavirus epidemic that was recorded in 2002, which was SARS-CoV, 774 deaths were recorded, but nearly 10% fatality rate. And the mean age of death was 50 years old. Then when we look at the next coronavirus epidemic, MERS-CoV, which really the hotspots for that were in South Korea and Saudi Arabia, 858 deaths were recorded, but the fatality rate was extremely high, 34.4%. And then when we compare that with the current pandemic that we're in right at the moment, we have far more deaths and globally, so no geographical hotspots. So really, this is really a global pandemic. And even though the fatality rate is between 1% and 6%, and this really depends on age, it really is in elderly people that we're seeing, or those over 65 that we're seeing this increased death rate. And if we look on the next slide, we can see that the actual death rate when patients are stratified by age is actually really high. So over 80 years old, the death rate is closer to 15%, whereas those under 50, it's between 0.5 and 1% as seen here. So we are in the middle of a public health emergency, primarily because of the number of deaths, the speed of spread, and at the moment, the fact that we don't have a vaccine and our inability to control this epidemic as it stands at the moment. So something that's been discussed in the media, in the general non-scientific media, but also in the scientific literature as well, is the R0 of COVID-19. So I thought I would just introduce this as a concept in case you're not familiar with it. So R0 is defined as the number of new cases that an existing case generates on average over the infectious period. And what this means is in reality is that R0 can be calculated by looking at the number of new cases over existing cases. Now, what this really depends on is our ability to define and confirm these new cases. And we'll talk about this more when we talk about testing. But what it means in reality is if R0 is less than 1, the disease will start to die out, because for every new case, it infects, that patient will infect less than one more person as they carry the infection. If R0 is 1, then the disease is stable, and if R0 is greater than 1, then the infection will spread. Now, the thing about R0 is that it can change over an infectious period in a susceptible population. And what is really influencing the R0 of coronavirus at the moment is the fact that we are all socially distancing and that we're in lockdown. So the less we see each other, the less likely we are to actually, for an existing case, to actually infect a new case. But let's think about viruses generally, not in the context of lockdown and not in the context of social distancing. These are R0 values of some more familiar viruses, if you like. And what I wanted to point out here is that Ebola, which we all consider to be highly infectious and an epidemic that was extremely hard to control because it wasn't really understood how this virus was working at the time in the context of where the epidemic was happening. Ebola R0 is 2, which means for every one existing case, that patient can infect 2 more, or on average infect 2 more new cases. Measles, which you will also be familiar with, has an R naught of 18. It's incredibly contagious, and we can talk about this in the context of vaccination and the public health strategies in the Q&A at the end of this talk. At the moment, it's thought, and even though this is a subject of debate, it's thought that SARS-CoV-2 falls somewhere between an R naught of 1 and 3. What is certain is it's more infectious than influenza, which has an R naught of about 1.2, but it's not really clear exactly how many people every new case can infect, because we don't have a blanket testing regime in place globally yet. So these are estimates, and this goes with a lot of the data that I'm going to show you today. So when we look at that in the context of other diseases in a slightly different way, what we've got up here is the case fatality rate. And one of the really key things about Ebola was that the R naught was estimated to be about 2, but the case fatality rate was extremely high, which is one of the reasons that the epidemic was so severe. When we compare this to MERS, so this is MERS-CoV, also the other coronavirus, the case fatality rate here was very high, and the reproduction number, the R naught, was actually varied between 1 and 6, depending on the public health and the cultural setting, and also the public health initiatives that took place at the time. You can see that COVID-19 down here sits really within the same region as seasonal flu, but is now thought to be actually more infective than seasonal flu. So this is data that was taken from a news article on the 23rd of March. So why have we ended up with a pandemic on this scale? And one of the reasons is to be that patients actually can present or not present, can be asymptomatic, but can be contagious for a considerable time before the onset of symptoms. And this is a really nice schematic that I actually got off Twitter, but is supported by a lot of data and some non-peer-reviewed, admittedly, but some clinical published. And the Twitter handle that provided this is ImmunoFever. And you can see that, as with any viral infection, exposure to the virus precedes symptoms, but depending on the virus, whether you're actually shedding viral particles or whether you're contagious really depends. You may be familiar with chickenpox, which has a very long incubation period of 2 to 3 weeks. So patients can actually be passing on the infection before they present with symptoms. And this is really a significant problem in a global pandemic where there is no global testing in place to see whether patients are positive and people are still moving around. So this is one of the reasons that the lockdown has been so successful in many countries. So what we're looking at here are weeks along the bottom, and time 0 is when symptoms start. And these are the symptoms that have been reported in the literature, and not every patient will present with all these symptoms. But the issue is that patients can be contagious, it's thought, for up to 2 weeks prior to presenting with symptoms, if they present with symptoms at all. And again, this is really something that hasn't been really well defined in the literature as yet. So how do we go about testing for the virus? In order to get some really solid quantitative data on the prevalence of the infection, the number of asymptomatic patients, the number of patients who've died who are positive, confirmed cases that may not be in hospital, we need a good testing regime globally. And one of the easiest and simplest ways to test for the virus is to look for viral antigens in either nasopharyngeal swabs or throat swabs. Viral particles have also been found in feces and in blood as well. But one of the most easy non-invasive tests is to use throat swabs or sputum, with a caveat that I will talk about in a minute. So this is what the coronavirus looks like. You may have seen pictures of this, called coronavirus because of the crown of the spike protein that envelopes the virus. It's a single-stranded RNA virus. And this is a really nice picture, I think, of the comparison between SARS-CoV, MERS-CoV, and then SARS-CoV-2. And it shows the genome assembly of the major antigens of this virus. Now, there's been a lot of talk about spike protein, and I'm sure we'll talk about this in the question and answer session at the end. But it makes other really key antigens as well: envelope antigen, a nuclear capsid antigen, and a membrane antigen as well. It also makes these polyproteins, ORF1A and B are shown here, but there are also other ORF polyproteins which can be used to identify the virus. So the idea of testing for the virus itself is to try and amplify the RNA that the virus actually has within its capsule. And as I said, you can do this from sputum, nasopharyngeal samples as well. So it's interesting to look at the slight difference genome assembly between these different viruses. And what is really important when we think about testing is to be able to show that if a patient, whether a patient is positive for SARS-CoV-2 or SARS-CoV, this is really important because the presentation of the infections are so similar. So we test for the virus generally using something called RT-PCR, which we would talk about more later. And this is a way of amplifying the nuclear material of the virus. Why do we want to do this? Because we want to know the viral load of patients. And this is a figure taken from a New England Journal of Medicine article that was published again towards the end of March, which shows that from nasal swabs in the top box here, throat swabs in the middle box, and aggregated values here in the lower box, you can see that viral load generally declines with a post onset of symptoms, which you can see here. So this is days. So that means that we can use this very simple test to see if a patient is gaining immunity or is starting to eliminate the virus from bodily fluids. This is the way most viral infections proceed. So the viral load does decline with induction of immunity. And there were only 15 patients in this paper, and that seemed to be the case with the patients that presented that were looked at here. However, what we still don't know is whether viral load correlates with clinical severity. It would be really nice if we could use viral load as a prognostic factor, very much like we do with HIV. So with HIV, we use the same process. So we have a PCR amplification test. And once the viral load drops below a certain threshold, we know that the patient's immune system will start to recover and the patient's clinical symptoms will start to not really resolve, but will decline to improve that patient's outcome. It would be great if we could do the same thing with coronavirus. What we still don't know either is whether the viral load is high or low in asymptomatic patients. And this is really important from a testing perspective and from an R0 perspective, from a public health perspective, in order to manage an infection globally. If the viral load is high in asymptomatic patients, is it because they have more immunity or better immunity? Or are they asymptomatic because their viral load is low? And this is something that we still don't understand. So, goes for flu, for hepatitis A, hepatitis B, and even HIV. You tend to have an asymptomatic stage once you've been infected before your immune system starts kicking in and you start to have symptoms. And this is the asymptomatic stage varies with viruses, and this is what's still uncertain with coronavirus. And then we have a period of onset of symptoms. In the context of coronavirus, this would be when patients start to self-isolate or present at hospital with severe symptoms. As far as the immune system goes, the first thing that happens is that IgM, which is an isotype, a type of antibody, becomes detectable. And this is the case with all infections. This is how the immune system responds to infection. Hopefully, then the patient starts to recover and IgG appears. Now, IgG is much more long-lasting and is the type of antibody that we would want to detect. It remains in the blood and can provide long-term immunity depending on the virus. So this is the reason that we're looking for a serological test for COVID-19. Antibodies reside in the blood of patients. If you take a blood test, you can then detect antibodies. If you can determine a ratio between IgM and IgG, you can start to determine whereabouts on this timeline patients are sitting. And if you do that in conjunction with viral load, you will know whether patients are still infected or not. The problem with a serological test is that it's retrospective. So if you think about that timeline, you have your asymptomatic period, you present with symptoms, and you need to wait for your immune system to kick in order to produce antibodies in order for us to be able to detect them. As I said, normally for infection, you would see IgM first and then IgG. IgM is usually indicative of an ongoing infection and usually indicates that the virus is still there, for instance, and IgG usually indicates immunity. But what we don't know yet is whether this is the case for SARS-CoV-2. And this has huge implications for testing, for immunity passports that a lot of governments have been talking about, and for trying to contain the pandemic as it stands at the moment. Now, I just wanted to share with you a couple of that peer-reviewed, but it does give an indication of why there isn't any certainty about either the role of antibodies in coronavirus yet, but also whether we can actually create a range or a threshold, as you would do normally with clinical diagnostic testing, in order to determine whether what you're seeing is indicative of immunity or not. So what we're looking at here are individual cases. So we only have 6 cases here. And this is, I think there were only 20 patients in this study where they did serological testing at different days after the onset of symptoms. So this does not include asymptomatic patients, but does include non-critical compared to critical patients. So what we're looking at here, firstly, and most importantly, is the RNA showing the presence of the virus. And we're going to talk about the tests a bit more in a minute. But what I want you to notice first is that in this lilac line, there are dips and peaks, troughs and peaks for the RNA detection. And this is really important in the context of understanding the prevalence of infection. And then in the red line, we have cumulative antibodies, so total antibody. We're not looking at coronavirus specific antibody with the red line. The blue line shows you IgM. And as I showed you in the previous schematic, we would expect IgM to come up at the beginning of infection and then decline and allow the IgG to come up and stay in order to provide long-term immunity. Now you can see here from these patients that this pattern does not persist in the non-critical patients at all. And we can't even make a correlate for the amount of antibody correlating with whether these patients are critical or not. When we compare this to the critically ill patients, and this is all on the same scale, you can see up here. So we've got the relative antibody in the y-axis, we've got the relative antibody concentration, and you can see that it varies completely. The conclusion that the authors came up with was that actually total antibody titer seems to be associated with critically ill patients. And this is just a snapshot of the patients that they studied in this, that they looked at in this study. But you can see that the IgM and the IgG seems to be low in these critically ill patients. Studies have been published but not peer-reviewed since this point, so this came out at the end of March, have not really borne this out. So there are critically ill patients who seem to have a lot of antibody and equally critically ill patients who don't seem to have a lot of antibody. There's also some evidence that asymptomatic patients don't make a strong antibody response. How do we detect these patients in the population if that's the case? Some patients have undetectable RNA in the upper respiratory tract. So these are nasopharyngeal swabs where we would hope to find viral antigen. Some patients seem to be negative for RNA but positive for antibody. And we also still don't know how long antibodies remain and whether they're truly protective in these patients. So how do we test for immunity then? If we don't, normally when we talk about vaccinations, what we're aiming for is a generation of memory cells that can persistently make antibody, which helps to neutralize the infection. What we don't have at the moment is very good evidence that antibodies generated during COVID-19 infection can neutralize the infection and provide immunity. But how we would normally look for these antibodies is using a technique called ELISA. Now, the majority of ELISAs that have been developed for serological testing have antibodies against receptor binding protein of the spike protein, which is the S protein I showed you on a previous slide. The FDA, or the Food and Drug Administration in the USA, have approved a Celex kit for use. The sensitivity is 93.8 and the specificity is 95.6. There's been an awful lot of debate about whether sensitivity and specificity is important in these tests. And I'm just going to give you two definitions, which we can discuss in more detail later. But sensitivity is the probability that a person tests positive if they are infected. And what this means for this particular test is that 6.2% of infected people will test negative. Specificity is defined as the probability a person tests negative given that they're negative. And this means that 4.4% of negative people will test positive. Celex themselves have also said that false positive results may occur due to cross-reacting antibodies. So when we're screening a large population with a low infection rate, the probability that you are actually positive drops in real terms, meaning that the error rate in the test is actually much higher. And we can talk about that in more detail. But what it means literally for you and me is that if you test positive for antibodies and you aren't actually positive, you will still be susceptible. And there's huge for actually controlling the spread of the epidemic and preventing a second peak or a second wave of infection. So there are other options for testing. And what I wanted to particularly point out is a neutralization assay. So we've talked about PCR and ELISA. A neutralization assay takes longer. But what it actually does is it tests patient antibodies in an immunological assay, which is a functional assay, which looks for reduction in viral growth. This is something that isn't online yet and something that would be very difficult to roll out as a global testing scheme. So my last few slides are about vaccine development, and we can talk about this again. But the first generation of vaccines that were developed particularly for virus were whole organism vaccines, live attenuated or killed vaccines, and many of the vaccines you're familiar with, such as measles or hepatitis A, fall into this category. But since then, using molecular technology, we've been able to create subunit vaccines, defined protein vaccines, and recombinant protein vaccines as well. And you'll have seen coming online vaccines for viruses such as Ebola, DNA vaccines, which can be made very rapidly, but have their own advantages and disadvantages. So as far as COVID-19 goes, this was a summary published in the New England Journal of Medicine of the different technological platforms that were being used to try and generate a vaccine against COVID-19. And you can see it covers the whole spectrum from inactivated right through to DNA and RNA vaccines as well. More recently, published in Nature Reviews in Drug Discovery, you can see that the pipeline of COVID-19 vaccines by technology platform is really based around recombinant proteins. And this is because we have the sequence of the virus, and it seems that antibodies against the spike protein may or may not actually be protective. Many of these vaccines are being developed in private companies with the caveats that hold, but this shows us where the money is for developing vaccines. Very few of the vaccines are being actually run in public institutions. So as a summary, I haven't talked at all about the immune response to coronavirus, and we can talk about this, about the severity of disease and how we can try and diagnostics. What we're looking at is detection of the virus, of the RNA of the virus by RT-PCR, and then trying to develop a test that has high enough sensitivity and specificity that it can be used in the general population where the prevalence of infection is as low as 3%. That's the final aim. Okay, thank you very much for listening. And over to you, Mushraf, for question and answers.
Thank you very much for the presentation, Jo. Very informative. You've covered a lot on the topic that we're going to discuss today. Now, I just need to note there seemed to be a little bit of a lag in the internet, just to let you know. So your presentation was very clear, but there was a bit of a delay, slightly, just to keep you posted. So let's start with the topic in hand. Just making sure, can you hear me, Jo? Is my voice
Yep,
clear?
I can hear you fine, yep.
So what are currently the standard tests used for detecting viruses? So not specifically SARS-2 or SARS-1. To detect a viral infection being DNA and RNA, what are the standard tests being used now?
So I would say that the gold standard is PCR, as I outlined in the presentation here, because in order to determine viral load, you need to know how much you've got, if that makes sense. And the best way to do this is to amplify the genetic material itself using PCR.
And is there a specific challenge with SARS-2 being that's a single-stranded RNA in detecting it, or does it fall within the same brackets of DNA and RNA detection?
Yep, I think that the techniques to detect the virus are pretty well established for viruses as well. And so the sticking point, the main challenge was to have the sequence of the new type of coronavirus, of COVID-19. But once that was established, once you have the sequence and the genetic sequence of the virus, then you can actually design. It's fairly straightforward to design a test, but then obviously it needs to be optimized and the sensitivity and specificity needs to be worked out.
Okay, so you talked about the specificity and sensitivity. Before that, you want to give us a brief about the PCR as a technique and how, while we hear PCR in the news being used as a technique for detection, it's not really PCR per se. It's a specific type of PCR.
Yes, it's reverse transcriptase PCR. So we're only amplifying the RNA of the virus, not the DNA of the virus. And the main challenge in designing the technique or designing the assay is that it doesn't amplify any human DNA or any other possible types of viruses. And because we know the genomic sequence and the genomic sequence of many other types of viruses, we can look to see if the bit that's amplified is very specific for the virus that we're interested in. The other advantage of the test is that because it's fairly cheap and fairly portable, it means that unlike the antibody test, we can actually look for more than one type of bit of genomic sequence, if you like, in the virus. So for instance, the FDA RT-PCR test for COVID-19 requires 3 positives across the genome of COVID-19 with all the associated controls in order to confirm that a patient actually does have the virus.
So we were very lucky, I'd say, in this pandemic. Looking at the dark picture, there was a bit of light because we managed to get the genomic sequence of SARS-2 very early when it started in China in the endemic. So Wu et al., Zhu et al. And Zhu et al. Were one of the first papers to grow out the rest of the world, the genomic sequence. Is this common within the scientific community? Do we usually manage to sequence the pathogenic genome early on, or has this been unprecedented in this front?
So I would say that a lot of lessons were learned from the Ebola epidemic. So in the first 2 weeks of the Ebola epidemic, 99 different samples, and that was unprecedented at the time. The technology has come on so quickly in order to sequence a whole pathogen, if you like. It's made it much more accessible to get the genomic sequence of a pathogen in a short space of time. And we should be extremely grateful to the Chinese scientists who actually made the COVID-19 genomic sequence publicly available in such a short space of time.
Yes, I mean, not only that, but the sequence of the S protein, spike protein that you've highlighted in your presentation is key, especially when we're going to brush on the vaccination process later on in the webinar. So I want to take you back to the slide you talked and you brushed up briefly on specificity versus sensitivity. So how is that applied in the PCR? So what do you mean by that?
So within PCR, it's much easier to get a very high sensitivity and specificity because you're looking at a unique genomic sequence. The problem comes when you're looking at antibodies, because antibodies can cross-react with other epitopes or antigens from either other viruses or other infections. And really, the best, even though you can engineer antibodies to recognize very specific epitopes, unfortunately, the reason antibodies work so well is because they do cross-react with various different epitopes. And that's one of the reasons that our polyclonal antibody response works so well. So with PCR, you can increase the sensitivity and the specificity of the PCR by testing for more than one region in the genome. Does that make sense?
It does. And it's going back to what you were saying about polyclonal antibody, how the great advantage of being able to cross-react works as a disadvantage in terms of testing and specificity at times. Can you highlight a little bit about the variation in samples? So you said that you've got samples that have low RNA with high serology and vice versa. How would you explain that? Why is that happening?
It's hard to explain from a technical point of view, but every technique has a threshold, a minimum threshold, and it's the same with looking at HIV, for instance. However, PCR is extremely sensitive, and in the case of coronavirus, what appears to be the case, because the infection tends, in critically ill patients in particular, tends to sit in the lower respiratory tract. And what means is that when you take nasopharyngeal swab, you're actually missing the site of infection. So that's one possible explanation. Another possible explanation is that these patients carry a mutated form of the virus, but that doesn't seem to be panning out with the data that's coming out in the literature. Mutated form of the virus doesn't mean a different strain necessarily, and actually the rate of mutation in coronaviruses is much lower than you would expect in other viruses, for instance influenza, but we still don't really know for this particular coronavirus.
When's the best time to take a sample for us to get the most optimized result? Do we know that yet?
No, and I think the best advice is to take as many samples as you can over a long period of time. So you'll have seen from the data that I showed you that the patients are constantly being sampled, particularly for viral load, to look at to see whether the load is coming down or increasing, particularly in critically ill patients, because it's not until we get rid of that virus that the bodies can actually start to recover. So I would say frequently, definitely from onset of symptoms, but to continue past recovery as well.
So in terms of the PCR not being able to detect, even though with the high sensitivity, or looking for a secondary type of test to confirm the PCR, what tests would we go for?
So I believe there are ways of checking for the functional aspects of the virus in vitro, but this takes a long period of time. It takes much longer to test for. So really, the best option would be to go for the site of infection in order to confirm that the virus is there. But then we can't exclude clinical diagnosis, of course. So when a patient presents with what very much seems like a COVID-19 presentation, requires oxygen, requires ventilating, then even if they're negative for the RNA, I would suggest that they probably still have the virus.
So histological samples, you're saying, when you mean at the site of the infection?
No, I mean taking, for instance, BAL, so bronchiolar lavage fluid. But that in these patients who are critically ill, that would be extremely difficult.
At that stage, of course, in the illness. Serology, IgGs. Where are we now, and what do we know?
Well, the ideal is to have a serology test that can detect both IgM and IgG, or have two parallel serology tests that detect both, so that you can determine a ratio for the patients. So I would suggest that having IgM is very important to see if the patient's actually making an antibody response. But then also looking for IgG is very important as well, to show whether the patient may actually be starting to have a protective immune response. There are several kits out there that different countries are using. So the Chinese have a kit that they're using from a very specific company. South Korea used a different kit. Germany used the Roche kit, and the FDA have approved the Celex kit. I'm actually not sure which kit the UK government has bought, but I do know that its sensitivity and specificity is not really sufficient for it to be used in the general population. Antibodies out there.
What do you mean by not so specific enough?
Apparently. So this is what I've heard. It's only media sources. Apparently the sensitivity and specificity is not sufficient for the reasons that I've shown on this slide. So if you're screening patients who all have symptoms, the chances of finding them correctly positive or negative are high. But as soon as you put that assay into a general population where the incidence of infection is low, your error rate goes through the roof, and you end up having a large proportion, a large number of people who will be positive, who will appear to be positive for the virus, but who actually aren't.
So IG tests appear to be not as reliable as PCRs, understandably, because you're looking at your immune reaction to the viral infection rather than looking at the virus itself.
I wouldn't say not reliable, because I just don't think we have the right test. It can take years to develop a really reliable test, and we have them for hepatitis B, for looking at vaccine responses. We have these tests, it's just that we don't have one yet for coronavirus, that's all.
And are we looking at agglutination tests, looking at immune complexes? Is that what we search for?
So I think agglutination tests really indicate clinical severity. But I don't know a lot about those. What have you read about agglutination tests?
So just from the principle of the test, by looking at immune complexes formed, either adding the antigen itself and looking if you have clumps appearing, or adding the antibodies to detecting the antibodies. It appears to us as lab specialists that agglutination test on the surface appears very easy to apply. But of course, knowing how hard it is to generate a proper, highly specific antibody adds a lot of respect to that technique itself. Would you say that we can use antibody tests to go hand in hand with PCRs, or does the timing of the increase of antibodies, either total antibodies or IgGs or IgM, can give us different indications rather than the presence of the virus, maybe the clearance of the virus?
No, I would say that doing them hand in hand is really important, and that shown this through my slides here. So you can see in many of these patients, in fact in all of them, even though this is a short timeline, so it only goes up to 25 days post onset of symptoms, but you can see that the viral load remains high, even in the presence of high antibodies. So to go back to your agglutination tests, that would definitely show you whether a patient is making antibody that's virus specific. And that's an important indicator of a healthy if you like, immune response in these patients, but what it doesn't tell you is whether that antibody is really protective.
So it can't replace it then? We can't say that antibody tests can replace PCRs because you've got a different fluctuation between what the antibodies are showing and what the viral load is?
Well, to go back to the HIV example, viral load is really key to understand the clinical outcome for patients. And so when you give them antiviral therapy and you see the viral load come down, it's known from hundreds of thousands of patients that then their immune system starts to recover. Now, one of the key problems with patients who are critically ill with coronavirus is that they end up with a lymphopenia, reminiscent, if you like, of HIV infection. And it's now known that coronavirus can actually infect T cells.
Yes.
What we really want for these patients is that their T cell repertoire, their T cell reservoir, if you like, recovers. Maybe we will only see this when the viral load starts to decline. So looking at viral load in conjunction with antibody tests gives you an idea of healthy immunity, which is really important in these patients, and maybe one of the indicators of critically ill patients is their inability to mount the correct immune response in that context. The other important difference is that with these ELISA assays, what we're looking at is antibodies to a particular protein. So the way these assays are set up, you need to have recombinant protein on the plate in order to detect the antibodies. And how you choose that protein in order to detect the antibodies also determines what you're looking for in that patient, if that makes sense.
Yes, that point about recombinant protein on the plate, can you repeat it again? Because I think the internet lagged for a second.
That's okay. So in order to detect specific antiviral proteins, antibodies, you need to give the antibodies something to detect in the assay. So every assay is based on a recombinant protein, if it's not whole virus, in order to detect the antibodies that can see that protein. Does that make sense?
Yes, it does.
So a lot of the assays that have been developed are based on the spike protein.
Yes. So that leads to my final question on this topic in terms of then can serology tests be used mostly in terms of environmental health? And in your answer, can you incorporate your understanding of how this has affected, how this pandemic has shaped our way or understanding of environmental health as well?
So by environmental health, do you mean public health?
Yes.
Like public health strategy?
Yes.
Yes. So serology testing, given a reliable test and a test that really tells us about immunity, would be extremely important in public health strategy. The concept of immunity passports is a fantastic concept. I was listening to Professor Paul Romer on the BBC the other day, who's an economist, who was talking about the balance between the economy and getting people back to work and also containing this pandemic. And serology testing is really key to that, in that if you can tell somebody whether they're immune or not, if we know they're immune, we'll have to work essentially. And that's a really key part of trying to overcome the other effects of this pandemic. But also serology testing is really important for understanding how patients recover from the infection as well. It's important for prognosis, which makes it important for medical infrastructure and how we actually plan to treat these patients and look after them.
I would imagine if we had such a passport or certificate, you can then, one, you might face legal issues in terms of sharing your medical information with the customs officer. Do you think there are legal implications there, or they might appear any?
Well, you could imagine how that would affect immigration, for sure. But if used in the context of viral load testing as well, you'd be able to determine whether somebody was actually currently infected. All it tells you, negative serology test, given that it's a true negative, would just tell you that that person is susceptible to the virus, not that they're carrying the virus, which is a very different thing. And actually then that brings us on to herd immunity and how important it is to have to determine the percentage of the population that needs to be immune in order to protect those of the population who haven't been exposed to the virus or who can't cope with the virus.
You've mentioned the H word now, Joe. It's a controversial town. Would you then expect that we would follow the IgGs then? So if your serology certificate shows that you've been exposed and you've formed the proper IgGs against it, would that mean that you'd know how you can trace the pandemic or the epidemic around a certain city or village through the IgG exposure? Would that sound accurate to you?
I don't think you could trace it in the same way as contact tracing works, but you could certainly determine the number of people who've been exposed to the virus, which is very useful. But again, that's with the caveat that those that may have been exposed and asymptomatic may not have a very strong antibody response. That's the first caveat. But the second caveat is that we don't know how long the IgG antibodies will last. So for influenza, we know that after vaccination, antibodies only persist for 6 months to a year. They don't last a lifetime, unlike the measles vaccination, which does last a lifetime.
So
after boosting.
With MERS and SARS-1, and I'm just bring the name out. I'm referencing, so in case the audience wants to come and chase me with machetes, they know I've referenced the paper. So Fatima Amanat from Immunity Perspective. So they found that it lasts for 2 to 3 years, the antibodies in SARS-1 and MERS.
Okay.
So not long lasting. Is that common for memory IgGs? Do they last for that long usually, or do you have a longer immunity presence?
It really depends on the virus. So as I said, for influenza, they don't tend to last that long, but there isn't, because influenza is changing every year. Seasonal influenza is slightly different because of the rate that it drifts with the mutations that happen on the outer coat, which means that every time we're exposed to a new seasonal influenza, we need a slightly different immune response. For that reason, we never boost with our influenza vaccine because there's not really any point. Whereas if you look at the measles vaccine, it's a different type of vaccine, number one. So it's a live attenuated vaccine. It has 3 boosts in children, which then last a lifetime. But the immune response has to be boosted in order to generate enough of a memory response to last that long. But it depends, polyclonal response, it depends on the antigenic variation of the virus. So, but I do think that you make a good point. So SARS-CoV is our best sort of predictor of what might be happening with SARS-CoV-2.
Yes, and you'll see that even in the production of the vaccine, we are relying a lot on what we know from our attempts to develop vaccines against SARS-1 and MERS as well, which we'll brush on later on. Can I ask you about the question that's been presented again and again in this webinar? How much in the immunity against SARS-CoV-2 are antibodies involved? So is it a cellular immune response that we are relying on in the primary infection? Because you've shown the paper of Zhu et al., where you see an accumulation build, peaking starting after day 7, then peaking after day 15 for IgM and IgGs. And you've mentioned the word of total antibodies. We do know that you got a cellular immune response generating earlier, facing the accumulation of the viral load. Can you elaborate on this point more? And can you explain what total antibodies mean compared to when they say total antibodies and then you've got IgM and IgG? Is that an antibody that's not specific for SARS-2?
Okay, so to get a cellular immune response, your innate immune response needs to kick in. You need antigen presentation, and neutrophils are extremely important when it comes to fighting infection at barrier surfaces such as the lung. But you need to have that cellular immune response before you can see antibodies. So the fact that we see antibodies means that we have a cellular immune response. But what's really seems to be common across the critically ill patients or patients who have a very poor prognosis is a reduction in T cells. Now, we know for antiviral immunity, we need a strong CD8 T cell immune response. And when this response declines, it has a knock-on effect on the viral load and also on the cytokine environment that is at this barrier site. So, for instance, in this case, at the lung. Now, when you see an antibody response, a total antibody response, what this is really reflecting on the slide that's still on the screen here is the assay specificity. So this is what we're talking about. We were talking about before, that in order to detect virus-specific antibodies, you need to put a bit of virus on the plate. So in the assay. So this IgM and IgG graph that you can see here in the blue and the green are indicative of antiviral antibodies, but they're only antibodies against the bit of virus that's on the plate. So measuring total antibody response is very important. Now, some of this antibody will be completely non-specific and will not be to do with the virus at all, but some of it may be, but that's yet to be seen. So cellular immunity is always important when you talk about an antiviral immune response, and it seems that lack of these T cells or reduction in T cells or lymphopenia correlates strongly with those with a poor prognosis and critically ill patients. So we think that the T cell immune response is particularly important in these patients, and that your total antibody response may be partly non-specific, but may actually include some specificity as well that we still don't understand.
Can it be associated with the probability of secondary infections due to the severity bacterial infections generating?
So possibly, but on this graph that I'm looking at here, so there isn't a lot of long-term data. I think the longest serology goes up to about day 65, which is pretty good considering where we're at the moment in this pandemic. But of course, with influenza particularly, influenza is often followed by a bacterial infection in the lung, and that will generate its own cellular immunity and its own specific antibody response. But I think the graph that we're looking at here, infection, what we're really looking at still is a patent acute viral infection, as you can see from the purple line here.
One more question about lymphocytopenia, just taking advantage of you being here. So some of the theories that have been presented before with previous guests, one, that we're missing the lymphocytes in the blood because there's a huge influx of the tissue. So we should look in the tissue as well to see if the lymphocytes are there. That's number one. Number two, with this new, with our new understanding of the presence of the ACE2 receptor on T lymphocytes and macrophages, in this highlighted in this beautiful review by Promit Chera in the Asian Pacific Journal of Allergy and Immunology, where they show that ACE is expressed on T cells and macrophages. So are we getting a decline of T cells that are infected, being attacked by CD8 cytotoxic T cells? Or is it because of the damage we see in lymphoid tissue? Which one of these three, or is there a fourth you would say makes more sense?
So for me, the primary problem with the patients, if we start at the far end of the spectrum with patients who die from this infection, it seems to be primarily because of complete loss of the lung barrier. So lung epithelial cells are our main barrier cell type, a single layer of epithelial cells with mucus obviously piled on top. Loss of this epithelial barrier is a terrible prognostic factor for any patient, no matter what their infection. Lung epithelial cells express the ACE receptor 2 that this virus binds to. New pathogenic mechanisms is to really hit the lung epithelial cells and remove the barrier. Now, obviously, once you start to degrade or damage that barrier, start the immune response, bringing in your innate immune cells and then your T cells as well. And because we really need CD8 T cells to remove a viral infection, these are obviously a key cell type. And a lot of the cytokines that are associated with both acute and chronic infection in coronavirus are associated with our CD8 T cells. We need the CD4 T cells as well to provide T cell help, both to make antibody responses and to generate these CD8 T cells as well. So I think what we call the tropism of this virus is really its ability to completely actually damage the lung epithelium is a really significant part of the immunopathogenicity of this virus, and is one of the reasons that we're losing so many patients to COVID-19 infection.
So there was a recent paper from Shen et al. Where they used the plasma from patients who recovered from SARS-2 to treat or to administer, I don't know what the technique was, into clinically severe patients, and they saw recovery. And they found neutralizing antibodies against SARS-CoV-2. So would that be an indication of how important the antibody response is, possibly upon secondary infection?
Yeah, that is really good correlative evidence that the antibody response can be protective. It still doesn't tell us whether which antibodies are correlates of infection, but it's a really nice way to show that antibodies can be used in this way. And the same thing was done with Ebola infection as well, if you remember.
And antibodies cannot always be good, right?
Well, in order to show whether an antibody is a correlate of infection, you need to show really what it's recognizing, and then you can test for it explicitly, which means then that your sensitivity and specificity goes up.
In terms of pathology, we've got cases of antibody-dependent enhancement of the disease. What can you tell us about that?
I don't know a lot about it, but what I do know is that there are examples, clinical examples of viral infections where actually having certain clonality of antibodies makes your prognosis worse. Okay, and dengue is a very good example of that. And as well, I think Ebola, also there are some examples of antibody-dependent enhancement. And so one of the debates in the literature at the moment is that if we don't design a vaccine sensibly with enough clinical evidence behind it, then we risk antibody-mediated enhancement in the general population.
And that has been a worry with spike protein, with S protein. So the previous attempts with SARS-1 have shown that when you develop an antibody against the S protein, sometimes you get protective effects, and other times you get detrimental effects, an infiltration of eosinophils, and possibly death of the lab animals.
I don't think that's really well worked out for coronavirus yet. It's not clear. Spike protein is an obvious candidate because coronavirus uses that for entry into some cells.
It appears to be the favourite candidate.
I would agree with that. Yes, I would agree with that. Yeah.
So
it doesn't mean it's the only one, though.
No, of course. So early on in the epidemic, it was the sequence specifically for it was fastly available. Lau et al. And Rapp et al. One of the first few papers that provided the sequence for the S protein. Why is it, Joe, that the S protein has been highlighted as the best candidate so far?
I think because of what we know about SARS-CoV. So the S protein, the molecular structure had already been worked out after the epidemic in 2009. And MERS-CoV also has an S protein. So I think building on the huge amount of literature to do with SARS-CoV, it was an obvious candidate to look to see if SARS-CoV-2 also used it for cell entry.
So, vaccines. Where are we? What do we know? What can you tell us about it?
Well, as I said on my slides, there are lots of different ways to approach vaccination. You've already mentioned antibody-mediated enhancement, which is something that is a huge note of caution, if you like, for developing new vaccines. You want to be able to is to take it pretty slowly as far as testing goes. You want to be able to test it in healthy volunteers, and you don't really want to fast track a vaccine when you've got something this contagious. You want to have a nice sort of closed experimental population, if you like. But unfortunately, in the context of pandemics, this isn't possible. And so, as we saw with the Ebola epidemic, it is possible to fast track DNA vaccines in particular to very specific geographical locations. It's possible to get licensing, it's possible to design trials ethically. We're not at that point yet with coronavirus, even though there are phase 1 clinical trials underway at the moment. I think that the majority of these, I'm just going to check back to the slide, the majority of these are the ones that have actually gone through to phase 1. You can see here, so this is the recent article from Nature Drug Discovery, shows that 2 are to do with inactivated vaccines, which is the most straightforward way to develop a vaccine. So you take the virus, you inactivate it, and then you present this, you challenge a healthy person with this inactivated virus in order to generate a polyclonal response. Now, many people would argue that that's the best way to generate a vaccine quickly. However, it's not the cleanest way, and it doesn't really stop this problem of antibody-mediated enhancement. So if we move right down to recombinant proteins or peptide-based proteins, molecularly, they're much safer to generate. They're easier to make on a very large scale, they're easier to store, and they're easier to administer, but they don't, unless they're very, it really depends on the design, but they don't tend to produce a polyclonal immune response, which is why understanding about the protective immunity to coronavirus is really important in order to understand the best way to design a vaccine. The real advantage with DNA vaccines is that if we think about influenza, for instance, seasonal changes in influenza are basically changes in the genomics of that virus. If you understand the genomics and you have a vaccine based on the DNA sequence of that virus, sequencing a virus is very quick. Generating a sort of surrogate package, if you like, to as a vaccine that allows expression of that very particular protein that you know is changing, gives you a rapid way to adapt vaccines in the same timeline as a virus adapting in the environment or in people, if you like. So there are huge advantages to using a molecular approach to vaccination. Using an inactivated virus requires growth of the virus. It requires category 3 facilities in order to grow them. It's expensive. It's not something that every country can do. It's not something that you really want to ship around the world, and it has to be stored correctly. So there are advantages and disadvantages to using each type. I think the main reason that these inactivated vaccines are in phase 1 clinical trial is because isolating the virus is the first thing that you do when you have a patient presenting. Isolating the virus allows you to sequence it, allows you to grow it, and allows you to study it.
In terms of the immunity generated between the different types, I mean, do we see different immunity between live viral vaccines, attenuated or subunits or DNA, or do we expect the same reaction to all?
So obviously with an attenuated vaccine or an inactivated viral vaccine, your body makes an immune response, makes an antibody response to every protein that it comes across. Now, the problem with an inactivated vaccine is that it shouldn't, or the good thing is that it shouldn't replicate inside you. But when a vaccine replicates, it naturally produces a different set of proteins. So an inactivated virus won't necessarily produce the whole set of proteins that a live infection would produce. So the advantage of using a DNA subunit or recombinant proteins is that you can identify the protein that's the most important in immunity and actually present that to patients and to the healthy population. So there are pluses and minuses. You would expect an inactivated virus to produce a polyclonal response, and you would expect that to be protective. But understanding more about the replication of the virus and the proteins that it's producing may actually help you to target immunity much more specifically with a recombinant protein or a subunit vaccine. Oh, there's a good example of that actually, tetanus toxoid vaccine. So tetanus actually produces a soluble toxin which causes a lot of the pathology associated with the infection. And so the vaccine is actually against the toxin rather than against the pathogen itself. So that's a really good example of how understanding how an infection works and what it does in the body can actually help you design an appropriate vaccine.
So to give the listeners a little bit of a brief of where we are in terms of the 3 types of vaccines. So I'm referencing Chen et al. In the current tropical medicine report. So we've got adenoviral vaccines that Johnson & Johnson has been trialing now. University of Hong Kong, an influenza vaccine that expresses the SARS-2 protein. So I'm guessing they're using it as a courier or an adjuvant. Then Codex is developing an attenuated vaccine as well. Now this is in terms of the whole viral vaccine. And according to the journal itself, it appears that they inherently stimulate TLRs for the response to the virus, which is quite interesting. They're specifically discussing TLR3, TLR7 and 8, and TLR9. Subunit vaccines are also being elicited as well, of using a viral surface protein to introduce as a vaccine. So a specific viral surface protein without any specificities on which viral specific protein on SARS-2. And they want to elicit an immune response against the S protein as well to prevent its binding to ASA2. The final DNA acid vaccine, now I do wish and hope that the listeners would go to the journal itself to read more about it. DNA vaccine showed promising results in the lab with animals, but again, has yet been licensed to be practiced further on. So it appears that all 3 major types of vaccines are being trialed at the moment. In terms of the timescale, I think everybody agrees now that it's not something that can happen over a day. It needs between 12 and 18 months because of the main clinical trial or hurdles, that they would say, that you need to produce. I think one important thing that we need to highlight, Joe, is how focusing the shift of the whole world on producing one type of vaccine has its own disadvantages, where they're already discussing shortages of other treatments because major pharmaceuticals are now focusing on generating the vaccine for SARS-2, while treatments for autoimmune diseases like SLE, rheumatoid arthritis, influenza vaccines are in risk of being shortened because the whole world is now focusing on SARS-2 only.
Yes, I think that's it's a real problem and something that governments themselves need to address, because obviously it's a different problem depending whereabouts you are in the world. So there's been a lot of discussion about the TB vaccine, for instance, the polio vaccine, that these, even though coronavirus is clearly a significant pandemic and is affecting hundreds and thousands, millions of people, actually the effect of a lot of these other diseases that have been that the World Health Organization has been trying to eliminate over many years. We can't take our eye off the ball in this context, but also we don't know how having coronavirus will affect these people's susceptibility to these other diseases as well. So there are lots of knock-on effects of, like you say, concentrating on vaccine production, which is clearly very important, but we shouldn't take our eye off the ball. Communicable diseases that are prevalent globally as well.
So the targeted population who are at the highest risk of getting SARS-2 are the elderly. Now, we have a specific issue with the elderly and vaccines, don't we?
Yes. So as you get older, your immune system also ages, and two really important things happen in the context of coronavirus. So as you get older, your T cell repertoire changes. So your T cells are your adaptive immune cells that respond to any new infections, and your ability to respond to new infections declines as you get older, but you still maintain a memory population to fight the infections that you've already seen. So one of the problems is, as you get older and your immune system gets older, and you're presented with a new challenge, you're asking your immune system to rise to the challenge, and it doesn't do that as efficiently when your immune system is older as when you're younger. And we see this really clearly with responses to the influenza vaccine. So estimates vary, but the efficacy, so the success, if you like, of the influenza vaccine in patients over 70 varies between 65 to 75%, which means that 25 to 35% of patients will not respond to the vaccine at all. It'll be useless in them. So when we design a vaccine for older people, it needs to be trialed in older people. We need to think very carefully about the adjuvants and about the proteins that are going to be expressed by the vaccine, if you like, or stimulated, or being used to stimulate the immune response.
So one more point, and then we'll open the panel for the questions from the attendees. I'm happy to say that we haven't seen more hands raised since the last rock concert I attended. So that's great news.
Oh dear.
We do face in the lab some issues or some limitations in how we study viruses. Do we have reliable animal models for influenza and respiratory disease infections?
So I'm actually not very familiar with the animal models for influenza. I believe that you can study influenza in mice, but as far as coronavirus goes, there have been a couple of non-peer-reviewed papers that have come out, obviously in the last month or so, which have shown that one of the best reservoirs for coronavirus is actually cats, and not mice as such, not rodents really, not dogs, but macaques as well. So primates, so important, but I don't know any more about it than that, I'm afraid.
So Bao et al., a very beautiful paper, was saying that even genetically modified mice, where we genetically modify the expression of ACE2, they only express mild symptoms with the coronavirus. So that does present a very obvious limitation. All right, let's take some questions from the audience then. So we have Ghaida. Hello, Ghaida, can you hear us?
Yes.
Hello,
Doctor.
Hello.
Okay. Thank you for great information. Nice lecture, mashallah. My question, you talk about IgG and IgM a bit specific and sensitive. My question, what about IgG ability? Okay.
I didn't catch that last bit. What about IgG?
Avidity, yeah.
Yes.
Okay. I have another question. People when have the infection with the COVID infection. After that, recover from the infection. Can be a reinfection happen or not?
Mm-hmm.
Are they immunized?
Okay, thank you very much.
Thank you, Ghaida.
Mm-hmm. That's okay. So obviously, IgG avidity is really important in the context of eliminating the virus. I haven't actually come across any studies at all looking at IgG avidity with coronavirus, but clearly that's important, but it's also, I would imagine, HLA dependent as well. I don't know if that really answers your question. Would you like to come back on that?
Let's see, Ghaida. Yeah, Ghaida, can you raise your hand so I can find you? When she does, I'll open the mic for her.
Okay, but for the second part of the question.
Oh, she's here. She's here. Sorry.
Oh, she's here. Okay.
Yes, Ghaida.
Yes. Yes, doctor. Yeah.
Did you want to come back on that about avidity? I don't know an awful lot about it, but and I haven't come across any studies looking at IgG avidity in the context of coronavirus, but it would clearly be very important in eliminating the virus.
What do you think, Ghaida, in terms of the avidity? What do you suggest in that front?
Okay, some doctors in another lectures say the Petro test or now use it for detected the severity of COVID. Use the IgG avidity, but I don't know what's the mean of the IgG avidity in COVID test.
Oh, okay. What's the meaning of it? So and
why use it? And why use it IgG avidity in the COVID test?
Okay, so when an antibody binds to an antigen, the stickiness is very important because it determines the time that antibody can bind to the antigen, but also its ability to form complexes and to actually remove the antigen in question. So if you have a very weakly binding antibody, it may actually affect your ability to eliminate an infection. But what I would say is that given that there's so much discrepancy and so many differences between different patients, that actually the amount of antibody being produced is clearly not consistent between patients. But you're right that the avidity of the antibody for antigen is something that hasn't really been looked at and may actually turn out to be very important. As far as the second part of your question goes with looking at secondary infections, I think the jury is out on that at the moment from what I've read. So again, this comes back to how long immunity lasts, whether patients actually completely eliminate the virus and their viral load declines to a point where we can say that they're no longer infected, whether there's any reservoir of virus in these patients, and whether they never cleared it in the first place, or whether the antibodies that they're generating are just not clearing the virus full stop. And I don't think that there is any real consistent data on that to help us understand.
Thank you, Ghaida. Thank you. Tarfa, I believe she's back.
Yes, hi. No, I'm on mute.
Sorry for the mute, Tarfa. It was
no, that's fine. So I'll just try to address some questions from the audience, Joe. So
okay.
Dr. Wissam have asked a question that says, do you think it's a good idea to look at T-cell profile using flow cytometry, which may indicate disease severity progress?
Absolutely. No, that's a really good idea. And there are some scientists in Manchester who are doing exactly that. So to get, lymphocyte phenotyping is a really well-established technique, as you know. And so applying that to patients during infection not only teaches us about the type of infection that's going on in the blood, the type of immune response in the blood, but also may be prognostic for these patients, that perhaps lymphopenia is because of T cells exiting into tissue, but I still think it's a really good clinical indicator of the immune response and prognosis as well, because of this massive association of lymphopenia with severe disease.
Another question from the audience. You have mentioned before the viral viral was detected in feces. So
yes.
They wanted to know more about that. We have talked about this actually in earlier webinar, but people keep asking about if it's a mode of transmission and how much the viral load is detected in feces. From Neda Jamjoom.
Yeah, so I read a really interesting article about SARS-CoV that was published back in 2004, looking at how at the expression of the ACE receptor that SARS-CoV uses to get inside cells and where it was in tissues in humans. And the primary sites of expression were lung epithelium and small intestinal epithelium. Now, you'll have seen from, let me just get that slide up again. Can you still see my screen, Tafa?
Yes, yeah.
Yeah, good, okay. So here, this really nice sort of schematic showing the connection between symptoms and exposure. You'll see that there are gut symptoms associated with a subset of patients. And so the paper that I was referring to about viral load in feces, it seems to be short-lived, but it's there. So it doesn't give you as consistent a reading as upper respiratory tract or nasopharyngeal swabs do, but it's definitely there, and it may be one of the ways that the virus is transmitted. But it goes up and down.
Not confirmed. Okay,
No,
but there's no papers yet. Okay, I would actually encourage, Werner is in the house. I would really want him to engage on the diagnostic kit in Germany. What are I would really want to know, because I know that they're testing now, and we want to know if there's specificity and sensitivity of the ELISA kits to SARS-CoV to have worked.
And I agree, Tarfa. I think Professor Muller should participate. I don't want to highlight too much.
So let's take one question from the audience and give the time to Werner to engage.
To muster up a very debilitating question where all of us can't answer.
Don't, Werner.
So we have
Antonella.
Yes, let's take Antonella. Hello, Antonella.
Hi, thank you so much, Joe, for an amazing presentation. I wanted to go back to the test with PCR. Could you please show the slide again where you show the IgG?
Yeah.
Thank you.
This one? Or the one with the patients?
Yeah, there were critically ill patients and non-critical.
Yeah. Okay, yes. So
I have to say, I'm not a PCR expert. But I wanted to ask first, do you know if that's a qPCR or a one-step RT-PCR?
It was a qPCR. I think it was a one-step, if I remember rightly, but I can confirm that. I can let Mushref know. So if you look on this slide, I think Mushref will make these slides available. On most of the slides, there's a hyperlink to the journal itself. So you can see in the bottom right, you can have a look, but I think it was a one-step.
Okay, so my question is, you show that in non-critical patients, basically you have a stronger immunoglobulin response, whereas strangely in the critically ill, the immunoglobulin response is lower. So again, I'm not expert in PCR, but I guess these samples come from the mucosal tissues, so either nasal, buccal, or did you say sputum as well?
So just to clarify that, the PCR samples are for viral antigen, and they are from sputum and upper respiratory tract samples, but the ELISA samples shown on here are serology. So they're from blood samples. And when you say there's lower antibody levels in the critically ill patients, this is really the IgM and the IgG specific, so the blue and green lines, not the red lines. Yeah.
Okay. my question, maybe it's a very crazy one. So I launched a quick Google, and I was looking for inhibitors of PCR. It is said that even immunoglobulins can present, of course, let's say if the PCR is done in plasma or blood, in there they will be present in higher quantities. But I was wondering, if you take a nasal swab and you have the mucosal tract there, we know that there are immunoglobulins as well, for example, IgM. So what's your view on these? And also, do you know if when the samples are taken from patients, do you know if there is pooling of the samples? Because of course, pooling all the sources might lead to an increase in the possibility of the detection. So I don't know if they do this.
Yeah.
Yeah.
So my understanding, and I've never done this particular qPCR, but my understanding from reading the methods is that you need to have a viral RNA isolation kit for exactly those reasons. So many, and the majority, so you can see here that the patients themselves weren't pooled, obviously, because these are individual patient samples, and these were not, multiple samples weren't taken and then pooled. So the majority of papers, all the papers I've seen, either use sputum or throat swabs or nasal swabs. And I think actually, oh no, it wasn't the same paper, but you can see that from this paper here. So they actually looked at viral load in the nose and the throat as well, but separately. So you don't tend to pool clinical samples in that way. Yeah. But many isolation kits, and they're used clinically as well, for detecting genomic material, use a column-based matrix.
Ich bin jetzt hier drauf. Sorry.
Yeah. Use a column-based matrix in order to isolate the virus. I hope that answers your question.
Yeah, it does. Sorry, and just a last point. Also, you show in those figures that when it's undetectable, basically it falls, let's say, beyond the 40th cycle.
Yeah.
So another question I had was, do you know what's the housekeeping gene they use? Because in my experience, I worked with epithelial cells from COPD patients, and sometimes we had to use different housekeeping genes that come a little bit later, because of course when doing the delta CT, if you use an housekeeping gene which is very strong, then your gene of interest will not be so expressed. But we have evidence of genes coming very late in the qPCR, and so maybe appropriate primers would need to be chosen.
Absolutely. So the CDC and the FDA have both approved a primer set with associated housekeeping genes, but I don't know what those are off the top of my head, but you make a very good point. So these assays have been set up with those controls and that optimization in mind. But you can see from the slide that we've got up there that the CT value, is not zero.
Yeah.
So when we talk about viral load in the same context as with HIV, then the thresholds have to be very clearly communicated so that clinicians particularly understand what we're looking at. So you do make a very good point. Thank you.
Great, thank you.
Thank you, Antonella. I think it's about time we get Werner Müller to speak. Professor Müller, can you hear us?
Yes, I can hear you. Yes. Can you hear me?
Yes, loud and clearly. As
Hello, Werner. Yes.
Hey, Joe. I really enjoyed your talk. Very good. Yes.
Thank you, Werner.
You can comment
How
about.
are you?
You can comment about the chairing later, Werner. You don't need to.
Okay.
So what do you know about I think it's the Roche serology test that is being used in Germany, is that right?
Oh, I think there's one test used from the north of Germany, from Lübeck. It's a small company, and that's being used in Germany. And but it's still under debate. So it's about, they say, 95% accurate. And the problem is the cross-reaction to the normal cold virus. So at this time of year, people still had a cold, and then one may get people who look positive in the ELISA, but they may just respond to a cold virus. And they get they tested a random sample of people, and there were about 3% positive for the test. So it depends very much which group of people one tests. If one uses tests with people which had symptoms, it's a different thing than if one just uses any random person and test any random person. Then you may pick up positives, which may have nothing to do with the current infection.
Absolutely, yeah. So exactly.
So antibody testing still, antibody testing is still, it probably takes a lot of time, a lot of controls, and there are many attempts to make it more specific for this particular virus.
Do you know anything about the kit used or about to be used in the UK? Joe brushed on it, saying that doesn't have a good rep so far.
So I only read what's on the news.
Yeah, me too. Yeah.
Yes, and it was not so. There was a kind of a negative, let's say, negative press about the kits which were used in the UK. But it's also possible that it's negative press because I think those kits were made for long-term. So they looked at IgG, and Joe, you have shown that IgG comes up late. And if one tries to use the test early after the infection, then it's negative. And so one has to wait long enough to pick up the people who are now positive for the antibody test. So I think it's maybe a combination of lack of specificity and the wrong timing when the test was used in the UK. So I don't think that the kit per se is completely wrong, but it's maybe a combination of bad timing when the kit was used at a larger scale.
Yeah. I encourage everybody listening to go to the paper that Joe referenced for the antibody response.
Yes.
It's a very good paper that shows the timing in quite elegant way.
And maybe I should add that there's another type of antibody response there which is important, and that is IgA.
Yes, I didn't mention that. That's a good point.
Yes.
Yeah, respiratories.
Yes. And because it's a mucosal response, so it's not picked up by the current ELISA tests, but IgA is also a very strong component in the antibody response against the virus.
Why is it not picked up with the ELISA? Is it hard to be?
Because they are built on. First it's the blood. They look what in the blood. In IgA, you look probably better elsewhere. And it's also in the blood, but it's not so easy to detect. And the ELISA kits are built on building blocks, which are used in other infectious diseases. And there you have either IgM or IgG. So it's just the way the reagents are available. So if you do in the laboratory, one can of course test more carefully, and then one can also detect a very strong IgA response. But it's probably not relevant for screening. It's more relevant to characterize the antibody response in a research setting.
Yes.
Yeah. It's not clear yet whether the IgA response helps with viral clearance either, I don't think, is it?
No, it's not clear. No, but it can be detected.
Yeah. Would you say it's more of a diagnostic detection then, the presence of IgA?
No. And I'm saying that because it points to the type of immune response you have. So not every immune response leads to IgA. And so the fact that you see IgA means that you have an immune response which also favors IgA antibodies.
Werner Muller's has done extensive work on IL-10. And we've seen IL-10 being mentioned as part of the cytokine portfolio generated in SARS-CoV-2. Yes. Can you give us a little bit about the role of IL-10 here? We know it's a regulatory, dampening, anti-inflammatory cytokine. So how is it now being presented in this phenotype?
So I think what you have there, you see if I look at the picture, you all have the picture in the middle, you have a term which is called cytokine storm. And the cytokine storm is an event where you have a lot of pro-inflammatory cytokines secreted. And I think not everything, so one important one I'm missing here is a tumor necrosis factor, TNF, which I'm sure is also produced there.
Yes,
but
it is.
And there's also IL-6. You can see IL-6 there. You see R1, interleukin 1 beta there, and IL-6, IL-1 beta, and TNF, mainly in TNF, are strong inducers of interleukin 10. So what one expects is that you have this strong increase of pro-inflammatory cytokines, and as a secondary response to these cytokines, you get a very strong upregulation of IL-10, interleukin 10. So it's probably in order, it's like with the antibody response, before you first have IgM, then IgG, and I think in the cytokine storm, you will have first the pro-inflammatory cytokines, followed by a counterbalance by interleukin 10. Okay.
Yes. So does it appears later in the infection as well?
Yes. Yes, it should be later, and it's probably the readout of a compensatory response of the immune system to counteract the cytokine storm. So that's known from when people study sepsis. So this looks probably very similar to a sepsis.
Yeah.
So in sepsis patients, late one sees very strong levels of interleukin 10.
Is it an indication of severity, Werner? Would you say at this stage, when it appears late? Is it too much too late?
It's too late because the pro-inflammatory cytokine storm is probably doing a lot of damage, and IL-10 is not there fast enough to dampen the cytokine storm.
So a good paper to implement what you were saying, Werner, by Huang and Wang from the hospital in Wuhan. It's in the Lancet, which covers the point you just mentioned, in case the audience want to go back and read more about it.
Okay.
Thank you very much, Werner. Thank you very much.
You're welcome.
And it's an absolute honor to have you with us here.
Thank you, Werner. Nice to hear you.
Thank you, Joe. Yeah.
Thank you. All right. So, Tarfa, are you there? It's always nice to talk to Werner.
It is, yes.
Hi, yes.
Thank you for encouraging Werner to join.
Yeah, I was so happy to hear Werner. Actually, there's an interesting question, I think one last question, because we're running out of time, from a PhD student in Sheffield. She said they're trying to identify the proteomics profile of S protein. So she wants to know if you think that focusing on protein target as a therapeutic target is a good, maybe faster approach while people are waiting for vaccine. So actually, I don't know any.
Just say that again.
So the question is, do you think focusing on protein target as a therapeutic target, is it good, maybe faster to overcome this pandemic? I don't know exactly what she means. I really encourage her to participate and ask the question.
What's her name? What's her name?
Hatoun Al-Amri. Sorry.
Hatoun, raise your hand and ask the question. I think if I understood the question right, if we target viral proteins rather than the viral body or the viral itself, is it better?
So I think if I understand correctly, I think there are two different things that she might mean. The first is, if you identify the proteins, can you then identify neutralizing antibodies to that protein and then design maybe better tests to determine immunity? That's one thing. And once you have proteins isolated, you can test all of them for their importance in viral replication and inducing various different types of immune responses. And that would be really important to understand more about the immune response to the virus for sure. But secondly, if you can understand what these proteins do for the virus, what part they cycle, and designing them as a vaccine is clearly very important. So looking sort of left of the spike protein, for instance, are there other elements of the virus which are important to generate an immune response? But that's really to do with vaccines. And I think in your question, Tarfa, you said differently to vaccines. So I'm not really sure that I've understood correctly.
We want Hatoun actually to elaborate on that, because she just sent a very brief question and she's here with us. I don't know. Can we hear you, Hatoun? Hatoun? She's unmuted, but I can't. Yeah, I think. Hatoun?
It's fine. We can get back to her. We can get back to her later.
Yeah.
So
can we allow some of the audience to some of the people who raised their hands.
I would really suggest to hear from your student.
Yes.
Yeah, Mushrif. He always come up with the excellent question. I really want to know what he's going to ask us.
All right, Joe. Now,
okay.
So a lot of good students are here. My students and yours, Joe. So we have Asil Hassan. What's your question, Asil?
Yeah, hello everyone. Can you hear me?
Yes, we
Yes, I can hear you well.
Okay, thank you for the lecture. I enjoyed and I learned much. I have two questions. The first one, I heard you mention about the lymphopenia, but I saw a result for a person who got infected with COVID-19, and he got also neutropenia and thrombocytopenia. What's the explanation for this?
Yes.
And the second question, what is the role of Th1 and Th2 in the infection?
Okay, so the first question, if I understand correctly, was that the patient had a lymphopenia, but a neutrophilia. Is that right?
Yes, and thrombocytopenia also.
Okay.
Thank you, Asil.
So the lymphopenia is indicative, just for the rest of the audience, indicative of a reduction in T cells, but the neutrophilia is an induction of neutrophils. So what has been seen in a lot of patients is that they have a very high white blood cell count, but actually the percentage of T lymphocytes is very low. And this is consistently seen across patients. So what you've described is what is often seen in patients. So you see this huge neutrophilia that comes out to barrier sites. And that's often seen with a bacterial infection as well as with a viral infection, because neutrophils are so important in managing infection at epithelial sites. Does that answer your question or not?
Yes.
Okay, so that was the first question. And the second question was, could you repeat that?
What is the role of Th1 and Th2 with the infection?
Yes, so antiviral immunity is very sort of traditionally associated with the Th1 immunity, which means that your CD8 T cells and cytokines such as interferon gamma and type 1 interferons are incredibly important in eliminating a virus. However, in the context of coronavirus, it's very much clinical severity is very much associated with this cytokine storm that Professor Muller was just talking about. And the hallmarks of this cytokine storm are both the innate immune response, but then also elements of the type 1 immune response as well. In Th1 is really important in antiviral immunity, but in the context of clinical patients, they often don't follow a textbook, okay? And that's the way it seems to be looking with coronavirus as well. So often you'll see a mixed Th1, Th2 in patients, even during an antiviral immune response during a dominant viral infection. And this can be sometimes to do with the presence of bacteria that can induce some elements of a type 2, but also you see the immune response trying to resolve and trying to bring other cytokines up late, as Professor Muller was just describing. So IL-10 coming up late in order to try and manage this massive cytokine storm. So what you're looking for is a strong Th1, but there will undoubtedly be elements of Th2 in these patients as well.
And will this Th2 be detrimental to Th1, Joe? Because we know it's a counterbalance on both sides.
So I haven't seen any reports of strong Th2 cytokines in acute infection, which is what you would hope to see. And there are other conditions where patients switch from a Th1 to a Th2, and that's not really understood why that happens. So I don't know, is the short answer.
So the immune hypothesis is that it can also be down to viral load. Is that anything we can entertain?
Absolutely. So the reason any immune response resolves is partly because the antigen's removed, and having a strong Th1 response will remove the antigen, or should remove the antigen, and help to control replication of the virus. But if this doesn't happen for whatever reason, if the antibodies are not protective, if their affinity and avidity isn't high enough, if you don't have a strong enough Th1 immune response and the viral load persists, then your immune response will persist and won't resolve as well.
Arfa, who will we choose? Pick a name for us.
So I think you would want to hear from Ahdab, your previous.
Yes, Ahdab, hello.
Ahdab?
Okay. Hi, do you hear me?
Yes, we do.
Yes. Hi, Joe. How are you?
Yes, yes. How are you? I'm good,
I'm
thank
very
you.
good.
I'm
Thank you
okay.
very much. We are very delighted to have you with us today. And thank you very much for your
Thank
talk.
you.
It was very clear and easy to follow. So I just want to go back to your PCR point. So we said that the golden standard for COVID-19 detection, PCR as a diagnostic tool. And also we do have chest CT as an important complement to RT-PCR test as well.
Absolutely.
But it has been shown that the sensitivity to detect COVID-19 for RT-PCR is lower than that of chest CT. And it has been shown that some patient tested negative for RT-PCR, but positive for chest CT. And then after, there was a case report, and after 4 or 5 days of admission to the hospital, their RT-PCR tested positive. So do you have any comment on that? And also, do you think that we are really in need for other biomarkers to complement this chest CT?
I think you make a really good point, and that's what I was trying to say earlier about the fact that clinical diagnosis is so key to understanding what's happening in these patients. So the chest CT scan is obviously indicative of, is partly to do with the prognosis of these patients, but also to look at their clinical presentation as well. As far as the RT-PCR positivity goes, there are a lot of elements which can vary to do with the assay. So what I've seen in the literature is that there are different kits available in order to look at the virus. So that's the first thing. So the possibility for not being able to detect the RNA and for it not being sensitive are that either technically it's not picking up enough of the virus. That's one possibility. The second possibility is that the site of swabbing isn't correct in some of these patients. Now, what's clear with coronavirus is that it sits in the lower respiratory tract. So on this picture here, it would be deep down in the respiratory tract, not in the throat and not in nasopharyngeal swabs. So one of the reasons patients may suddenly become positive for the virus may be because of the coughing and the other elements of the clinical presentation, which means that the virus can now be seen in these throat swabs up here. So I think with any assay, it's difficult. You need to have the virus there in order to detect it. So if it's not at the site that you're swabbing, then you won't see it. And ultimately for the patient, the most important thing is to understand the clinical symptoms, which is why the CT scan is so important.
Okay, that's great. So I do have another one, if you allow me. So this question is for both you and Werner. It has been because about AL10, and my MSc project was about AL10. So I would like to ask about this question. So AL10, yes, we all know that AL10 has an anti-inflammatory effect, but it has been suggested that AL10 has also an immunostimulatory effect, especially on CD4, CD8 T cells, and also on NK cells. And also it can induce interferon gamma production. So do you think the AL10 that we see on COVID-19 patients, it's because of an immune stimulatory or anti-inflammatory effect?
I'm going to let Werner answer that, but my feeling is that I agree with Werner's initial assessment when I was talking just before, in the IL-10 is probably coming up to counteract the very strong TNF-alpha signature that is seen in these patients. And actually, there's been a big push to think about monoclonals to TNF as a treatment for these patients. And I think that very much reflects the anti-inflammatory role that IL-10 is thought to play in these patients. But as with everything else, time will tell, because I don't think we really understand that. What do you think, Werner?
So I think there are two, probably in this system, there are probably two cell types producing IL-10. One is probably the innate cells, which are responding directly to the TNF or IL-6 in that case. And that is what we see. The second is that at least if they have a very strong Th1 response, people have shown that later we have also gamma interferon producing T cells which start to produce IL-10 later. So I expect, I don't have any experimental proof in this case, but I expect that also the T cells, the Th1 cells from the response start to produce IL-10 later in the response. And so one is from the adaptive side of the antiviral response, and the other one is from the innate side of the antiviral response.
What about macrophages?
In terms,
macrophages.
the macrophages are the ones responding to the TNF. So the macrophages are producing IL-10 upon TNF induction.
So in response to TNF.
In response to TNF. So they first make TNF and then they respond to TNF. So they can do both. Yes. So there are also very strong IL-10 production by macrophages as well, depending on the way you induce it. Also, bacterial responses would induce a lot of IL-10 in macrophages later.
Would you say the bacterial response, then again to the point I've been raising with in previous webinars, and as I brought it up with Joe, is the IL-10 due solely to the viral infection, or are there elements of secondary infections through bacteria that induce the IL-10 as well?
Yes, so I mean, people have looked at complications in the viral response, and then there are signs of complications due to co-infections, particular bacterial co-infections. So people have seen that you have, let's say, also an increase in bacterial infections during the course of the viral response. So that could then play a role. Huh?
There's activation of TLR4 they've seen in bacterial infections as a co-infection. And
because I think we have all bacteria always around, and if then the immune system is challenged by the virus, then some bacteria may start to grow or to expand due to the change in the immune system, and then on top of the viral response, you also have a bacterial challenge, and also fungus, by the way, could also be fungal infection as well.
Yes, yeah.
It's interesting that you say that, Werner, because procalcitonin, which is a bacterial infection marker, is a marker of severity.
Yes. So maybe I just make one comment to Joe's anti-TNF suggestion.
Pleasure.
With pleasure. You've not done that before to us.
No. It's I mean, what people are trying, I think what people are trying in the case of the corona infection is anti-IL-6.
Yes, definitely. Yeah.
And not so much anti-TNF. And I think the reason is because the IL-6 has probably a more diverse role both in the innate and the adaptive immune response.
Yeah, and it's very much correlates with clinical severity as well in this cytokine storm phase. Yeah.
Yes, and so what people have measured is so there's a group of proteins induced by IL-6 which are called acute phase proteins. And then during CRPs, and then in the when the infection becomes severe, then one sees a very strong increase in the CRP response. And that is also, so you discussed about how to predict the outcome of the infection, and there have been also now some let's say observational studies where people have measured the CRP response in patients and correlated to the outcome. And if patients have a very high level of CRP, then the outcome is usually bad. So it's a bad prognosis marker now for the outcome. It's so, and IL-6 is being tried but it's too early. I mean, one should say there are a lot of clinical trials, and it's not all of them are done in a way that they are conclusive at the end. So one just has to watch and see,
and wait,
and wait and see. And it's very difficult at the moment to distinguish a good clinical trial from a trial, because people are just desperate. They try everything, and then you find good numbers. But if you then look at the patient cohorts, it's very difficult to know what would have happened to that patient cohort without treatment. So it may look good, but if you have the wrong patient group, that you have an outcome which would not be different from the control group, because it's so diverse depending on the specific makeup of the patient group which is studied. So it's very difficult to know.
On the heel of that, Werner, I'd like to also refer the audience to Promichera's review, where he discusses IL-6 in more detail.
Yes.
Thank you, Werner. Thank you very much.
You're welcome.
Thank you. Tarfa.
Okay, so interesting point. Everyone is talking about IL-6. Actually, I wanted to ask one question to Joe before we wrap up. So now we know that the source of IL-6 is mostly macrophages and maybe DCs. What other cells do you think is the source of this cytokine storm that caused only by IL-6 and causing immunopathology in the lung of critical patients?
Well, I think, as Werner said, T cells do make a lot of the cytokines that have been associated with clinical severity in these patients, but we shouldn't underestimate the lung epithelium in all of this. So they're clearly being infected by the virus and being damaged by the virus. So a lot of the chemokines that you can see on this picture here will actually be made by the lung epithelium. So I think it's hard to say. I think the conclusion is that there isn't one cell type that's making IL-6 and there isn't one cell type that's making chemokines, but it's the whole of the immune response working together, the innate and the adaptive cells, to generate this cytokine storm that's so clearly associated with severity.
Tarfa, if you would allow me, I have one of my students who wants to ask Joe a question. Joe, thank you for giving us the time. I know we've dragged this so long.
It's okay.
Abbas, are you there?
Yes, I'm here. Hello.
What's your question?
Can you hear me?
Hello. Yes.
Hello, everyone. Hello, Dr.
Hello.
Joanne. It's a great opportunity attending this webinar. Your presentation was amazing.
Thank you.
Abbas, just one second. Joe,
Okay.
I threatened him with marks if he didn't say that.
Yes, he did.
Bribery always works.
He surely did. My question is there any test done on T-cell exhaustion or colonial exhaustion in case of coronavirus? And what does it indicate to us? And how does it relate to what you said earlier about how we don't know how long does the IgG response take?
Okay, so I'm going to go back to one of my slides about the test. Spend time on the slide about the neutralization assays. So these are all to do with antibodies, but looking at T cell function in these patients is really important because we don't know why there's, or at least I can't tell from the literature yet, why there is this lymphopenia, whether it's because the T cells are exiting into the tissue, whether they're dying, or whether they're exhausted. And I think from a diagnostic point of view, it's probably not that useful to know if a patient's T cells are being exhausted. But what is useful is to use it as a prognostic factor, but also to think about what the virus is actually doing to the adaptive immune response. If you can prevent T cell exhaustion in some way, then obviously you can completely try and improve the prognosis of these patients. The likelihood is that it's persistence of viral load that actually induces the T cell exhaustion at this point, because you'll probably already know that the lymphopenia is the second wave of the infection. So it's in getting into the chronic stage of infection rather than the acute stage. So it seems to be associated with persistence of viral antigen, which would fit with what we already know about the immune response. But it's a very good point. Understanding the T cell immune response is really key to understanding the immunology of this infection per se.
Very good question, Abbas.
Thank you.
Excellent question.
Very good question. I may add, if you guys are interested about, the listeners, if you're interested about T cell exhaustion, there's a beautiful paper in Cellular Molecular Immunology by Zheng et al., where they showed the NKG2A, which is an inhibitory marker for NK cells and CD8. They are expressed highly in exhausted T cells and CD8 T cells and NK cells. Nicely written paper. I advise everybody to go and check it out. That can elaborate more. What do we mean by a T cell exhaustion? Do we see an increase in inhibitory markers? Is that solely what it is?
So the T cell stops responding to the signals it would normally respond to in order to proliferate, in order to clonally expand, and then to be able to help B cells and CD8 T cells in this case. So exhaustion is not necessarily the same as having an inhibitor on the surface of the cell. It's not the same thing, but it can result in the same thing.
And is it always associated with the drop in numbers? So the less cells you have, the more exhausted they are?
No. I would say an exhaustion is a function of a T cell rather than, and it can lead to apoptosis and loss of that cell altogether, but you can have a cell present that's exhausted and non-functional.
I see. Okay. All right, Tarfa, do you have any more?
So there was a question actually about the role of IL-1 in COVID-19, and it says if we can control IL-1, we can control, IL-6 if we controlled IL-1.
Mm-hmm. I don't know a lot.
So I think, I would really, I think it was mainly for Werner, IL-1, the IL-1 guy.
Yes, good.
So I don't know. This is I think most of the questions, and that's
okay.
it.
Well, Joe, I think we've taken a lot of your time.
I've really enjoyed it. Thanks,
yes,
Misha.
You've been very generous with your time to us. I mean, what else can be said that I haven't said to everybody I know about Joe? She's one of the most influential women I know. And she was my supervisor. She's a good colleague, but she's always a great friend of mine. And you can see from the love and the questions that have come from my colleagues how much she means to them. So we appreciate you, Joe. And most importantly, I appreciate the people I got to know through you. So thank you for your time.
Thank you, Misha.
Thank you for every second and every minute you gave my colleagues and my students as well. Any final words?
Problem. Thank you very much for having me. I've really enjoyed it. I enjoyed preparing for it, and I've enjoyed the questions too. It's actually given me a lot to think about.
And that's the drive. Tarfa, any final words?
Thanks a lot, Joe. It was lovely seeing you today, and it was an interesting talk.
Good.
Thanks a lot.
Joe, thank you very much.
Thank you very much. Thanks, everybody.
And thank you for Werner.
Thanks, Werner. Yeah.
Thank you, Werner, for joining us and complicating our lives. Thank you very much. Okay. Thank you very much. Thank you very much, everybody.
Thanks, everybody.
Take care. Bye-bye.
Okay.
Bye.
Bye.
Bye.